Y bueno, vamos a dar inicio a este nuevo seminario. Le pediría al doctor Luis que nos ayude con la introducción de nuestro invitado el día de hoy. Gracias a todos. Adelante, Luis.
Gracias, Carlos. Bueno, pues es un gusto tener a Sergio Grinstein con nosotros. Debo de contarles una breve historia de por qué surgió que yo lo invitara. Porque hace como unos quizás 3 años, o sea, buscando literatura sobre fagocitosis, porque yo no sabía nada de eso, busqué nada más en saber quién había publicado cosas sobre fagocitosis y macrófagos, y lo encontré porque fue quien en varios artículos que él escribió me orientó a qué se sabía y qué no se sabía. Entonces lo contacté en una ocasión para hacerle algunas preguntas más específicas y ya, pues yo pues como habrán visto se llama Sergio Grinstein, entonces el apellido Grinstein pues no suena de inmediato latino. Y entonces, pues para mí, pues era alguien pues canadiense, de nacimiento, quiero decir. Y pues fue gratamente, me enteré en ese momento de que él es mexicano, o sea, estudió su doctorado en México y ya después de sus estudios en México se fue a hacer un postdoctorado en Canadá, luego estuvo en Zúrich, y finalmente ahora es un investigador muy destacado en Canadá. Se encuentra en el Sick Children en Toronto, en el Hospital Sick Children de Toronto. Ha recibido muchísimos premios, tiene más de 500 publicaciones en revistas destacadas. Y pues su tema es señalización celular, varios aspectos en este ámbito relacionados, muchos de ellos, si no es que todos, en inmunidad innata. Y pues alrededor de este tema es que nos va a hablar hoy. Muchas gracias, Sergio, por estar aquí. Es realmente un placer, sobre todo, pues conocerte, porque pues, para mí ha servido para orientarme en un tema que me era desconocido. Adelante, Sergio.
Ok. Muchas gracias, Luis, por la introducción y gracias por la invitación también. Y, a pesar de que el seminario inevitablemente va a ser en inglés, porque la ciencia la hacemos en el laboratorio en inglés y las diapositivas están en inglés, no quiero invitar a la gente que haga preguntas, ya sea en inglés o en español, y quería hacer notar que la presentación van a ser 2 temas diferentes pero relacionados y diferenciados de la plática. Si la gente quiere al final de la primera sección hacer preguntas, con todo gusto interrumpimos ahí, si no esperamos hasta el final. Así que voy a compartir la pantalla. Vamos a ver. Okay. Perfect. Muy bien. So the topic I want to deal with today are 2 pathways, 2 proteins of pathogens that affect the biology of the host cell, and 2 cases of remarkable convergence evolutionarily of functions in bacteria and in mammalian cells. And just as a general reminder, when we are infected by pathogenic microbial threats, it's normally the first line of defense, which are cells of the innate immune system, macrophages, neutrophils, dendritic cells that deal with these pathogenic bacteria. And normally, macrophages, neutrophils, and dendritic cells can internalize pathogenic bacteria or other microbial organisms, deliver them into lysosomal-like organelles where they degrade them and break them down. But there are an important subset of bacterial pathogens that have evolved strategies to survive inside the host cells. Some of them escape the vacuole, the phagosome, where they're taken up, and grow unrestricted in the cytosol of the host cells. Others have managed to modify the structure of the vacuole to equally survive inside the host cell. And these are not a small family. These are not inconsequential. In fact, many of the primary pathogens that humans deal with are intracellular pathogens, and they include Legionella pneumophila, which is the causative agent of Legionnaire's disease, Chlamydia trachomatis, which causes blindness, Mycobacterium tuberculosis, which produces tuberculosis and kills still millions of people every year, Listeria, which produces gastrointestinal disorders. But the one we're going to deal with today is Salmonella. And Salmonella is a gastrointestinal pathogen. It produces bacteremia, arthritis, typhoid fever. It's really a major, major problem in the gastrointestinal pathway. And like some of the ones I mentioned before, Salmonella is an intracellular pathogen attacking primarily epithelial cells of the intestinal lining. And Salmonella enters the host cells actively. Unlike phagocytic cells that take up bacteria in order to destroy them, epithelial cells are infected actively by the bacteria. And what I'm showing you here is on the left, a diagrammatic scheme where we show how the bacteria ruffle the ends of the plasma membrane of the cells and gain access to vacuoles intracellularly. But this should become much more obvious if you look at the movie to the right, where we have labeled the plasma membrane of an epithelial cell with a fluorescent marker, and we're going to expose this cell to red-labeled Salmonella. And you can see the bacteria landing, and I hope this goes pretty fast, and I'll show it again in a second. But you will see that in this area of the cell, the bacterium lands and induces enormous amounts of ruffling, and the bacterium gains entry into the cell. So this is a very proactive mechanism initiated by the bacterial pathogen. And the bacteria do this by injecting a number of effectors into the host cell through what's called a type 3 secretion system, which is nothing but a macromolecular needle that traverses the membrane and the cell wall of the bacterium and also pokes across the membrane of the host cell. And once it reaches the other side of the host cell membrane, it injects a number of proteins that act as effectors. And in the case of Salmonella alone, there are upwards of 40 different effectors that the bacteria inject into the cytosol. Now, I don't want you to panic. I'm not going to deal with all 40. I'm going to concentrate exclusively on one of them, which is called SUP-B. SUP-B is one of the essential effectors that promotes entry of the bacteria into the host cell. And it does that, curiously, by manipulating host lipids. What we knew, and this is about 20 to 30 years ago already, is that SUP-B, the Salmonella effector, is actually a phosphoinositide phosphatase. And as a reminder, phosphoinositides are phospholipids that have a head group of inositol rings that can be phosphorylated in positions 3, 4, and or 5, attached to glycerol and has 2 fatty acyl chains. But in addition to being a phosphoinositide phosphatase, sub-P was known, and Salmonella were known to affect kinase activity inside the cell. And in particular, the activation of the AKT pathway, the AKT pathway being important in cell signaling and in cell proliferation. And what I'm showing you here is the phosphorylation of AKT in cells infected with Salmonella. You can see that in uninfected cells, AKT, which is present, is virtually unphosphorylated in positions 308 and 473. But shortly after infection with Salmonella, you get a massive phosphorylation of AKT. Now, this is not trivial at all because phosphorylation of AKT promotes host cell survival, and those provides a fertile ground for the bacterial to grow. It counters the pro-apoptotic and apoptotic nature of secreted factors, dampens the inflammatory responses. It expands the invasion targeting of cells, and importantly, it promotes oncogenesis. There are, in fact, populations in India that are chronically affected and infected by Salmonella that get cancer of the gallbladder as a result of the infection with Salmonella. So how does Salmonella induce the phosphorylation of AKT? Well, AKT is activated by phosphoinositides, PI34P2 and PI345P3. And in fact, we know that the activation of AKT, the phosphorylation of AKT by Salmonella, depends on sub-P, because if we generate a mutant strain of Salmonella lacking sub-P but having all the other effectors, you get normal infection, normal invasion, but no phosphorylation of AKT. So sub-P induces the phosphorylation of AKT, but sub-P is a phosphatase. So how does a phosphatase lead to activation of AKT? So over the years, people have assumed that the phosphatase activity of sub-P somehow mysteriously promotes the activation of a PI3 kinase. And it's not clear whether the PI3 kinase is of host or bacterial origin. And in fact, the lipids that are being degraded by sub-P that presumably induce the activation of AKT have not been identified either. And again, it's worthwhile reminding you that what normally activates AKT and AKT phosphorylation is an increase in the concentration of PI34P2 or PI345P3. And for years, we were unable to measure the generation of PI34 because there were no good molecular probes and because chemical methods were not sufficiently sensitive to detect PI34P2. But a couple of years ago, our colleague Jerry Hammond at the University of Pittsburgh developed a beautiful new probe that can detect in intact live cells the presence of PI34P2. And what this probe consists of is 3 tandem domains of the protein called TAP1 that are known to bind PI34 attached to a green fluorescent protein. And when this is expressed in the cytosol of mammalian cells, it will be floating in the cytosol until such time that PI34P2 is generated in the membrane, at which time the probe will bind to the phospholipid and will bring with it the fluorescence of GFP. So you look at a normal cell transfected with a probe for PI34P2, you will notice that there's a very faint level of cytosolic fluorescence with virtually no fluorescence in the membrane because at rest mammalian cells have virtually no PI34P2. But this is again a movie, and I'm showing the movie both in black and white and in pseudo color, the same movie, so you can look at either one of them. And what we are doing here is, as we did before, infected the cells with Salmonella typhimurium, expressing a red fluorescent protein. And the movie starts just before one of these bacteria injects SOP-B into the mammalian cell. And I think the movie is pretty self-explanatory. I think it's obvious that when one or two of these bacteria make contact with a cell, you get a massive recruitment of the probe to the site of the infection because you've locally generated a huge amount of PI34P2. If we use another similar probe to measure PI345P3, we get a very, very small response. So the primary product of the infection with Salmonella is PI34P2, this guy here. Okay, so is it SOP-B that's doing the generation of 3,4? And what we have here again is an image of PI34 being generated at the site of infection when cells are infected by wild-type bacteria. This is what happens when the cells are infected with bacteria lacking specifically the gene encoding SOP-B. Clearly, the generation of PI34P2 requires SOP-B. You can quantify the data, and it's really black and white. You don't need to be a statistician to tell that these are very different. So we know that SOP-B is required to increase 3,4P2, but is it sufficient to increase 3,4P2? And the problem with infection with cells is that there are 40 or so different effectors. If we want to isolate the effect of SOP-B, even though we know it's required, we need to deliver SOP-B to the mammalian cell in the absence of all the other effectors of Salmonella. Instead of knocking out all the other effectors, it's much easier to transfect the mammalian cells solely with a plasmid expressing SOP-B in the absence of bacteria. And so we are doing an experiment here where we transfect the probe for 3,4P2, and you can see again in resting, otherwise untransfected cells, PI34 is non-existent and the probe is soluble. If you now co-transfect the probe with SOP-B, you see marked generation of PI34 in the membrane of the cell, in this case throughout the cell, because the SOP-B is now expressed throughout the entire cytosol. And I think it's very clear that there is marked generation of PI34P2 induced by SOP-B, and that SOP-B is sufficient to do it. And in fact, we know that SOP-B has a critical domain following a cysteine, and that this cysteine in this critical domain is essential for the phosphatase activity of SOP-B. If we mutate this cysteine, which is positioned 460 for serine, we inactivate the phosphatase activity, and we also inactivate the ability of SOP-B to generate PI34P2. Here's the mutant. This is the wild type. Okay, so we now know that the expression of SOP-B is not only necessary, but it's also sufficient to generate PI34P2. Now, the problem with transfection experiments is that it requires many, many hours. And we don't know if the effect that we're seeing of SOP-B is a direct effect, or if it requires the induction of another intracellular pathway, such as the activation of a kinase, etc. And so, can we instead induce SOP-B mediated PI34 acutely without having to wait for the many hours it takes for expression by transfection? And for this, again, we collaborated with Jerry Hammond. And what Jerry did is he made a recombinant plasmid of SOP-B where, very close to the cysteine that is critical for activity, he introduced in this lysine hydroxycoumarine. When it's covalently attached to the lysine, hydroxycoumarine, which is a large molecule, blocks entry to the catalytic active domain of SUB-P. So this is an inactive form of SUB-P. However, hydroxycoumarine is very light sensitive. And if flashing light at 405 nanometers, we are able to dislodge the covalently attached hydroxycoumarine, release it from the site, and enable entry of substrates into the catalytic domain. And so photoactivation enables us to test the activation of SUB-P in a matter of seconds. And so what we did in this experiment is co-express hydroxycoumarine-bound SUB-P together with a PI3/4 biosensor. Before flashing light, SUB-P is inactive. And for that reason, there is no PI3/4P2, and the probe is soluble and doesn't know where to go. This is time zero. And we're going to start the movie, and you'll see what happens when we flash light and release the active site of SUB-P. I think it's a looping movie, but I think it should be very clear that upon photoactivation of SUB-P, the plasma membrane is now able to recruit the construct, excuse me, which is an indication of the formation of PI3/4P2. So the conclusion of these experiments is that SUB-P is acutely and sufficiently able to generate PI3/4 in the membrane of mammalian cells in the absence of any other bacterial effectors. So what we know then is that there's no detectable PI345P3 in the uninfected cell. And for that reason, the phosphatase activity of SUB-P cannot directly account for the formation of 3,4P2. So how do you form 3,4P2? It cannot be the phosphatase alone. Could it be stimulating an endogenous PI3 kinase of the cells? And so mammalian cells have three different kinds of PI3 kinases. The major type is the class 1 PI3 kinase that phosphorylates PI45P2 in position 3 to yield PIP3, which then presumably through the phosphatase activity of SUB-P could generate 3,4. If that's the case, the phenomenology that we're observing should require class 1 PI3 kinases. So is SUB-P stimulating class 1 PI3 kinases and then breaking down the product into PI3/4P2? Unfortunately, class 1 PI3 kinases have been studied extensively because they're important in proliferation and cancer, and they're very good inhibitors for class 1 PI3 kinases described, including wortmannin. And as should be obvious from these images, the formation of PI3/4P2 in the ruffle of salmonella-infected cells is completely unaffected by wortmannin. And in fact, not only wortmannin, but PI103 and another compound called GDC941, all of these are extremely specific, potent inhibitors of PI3 kinase class 1. They have absolutely no effect on the generation of PI3/4P2 by SUB-P. So clearly, class 1 is not involved. Okay, so it's not class 1. How about other types of PI3 kinases? And there's another type of kinase in mammalian cells called class 2 PI3 kinase. This one can phosphorylate PI4P on position 3 to generate PI3/4P2. Conceivably, SUB-P could be activating class 2 kinases. Now, unlike class 1 kinases, there are not too many or not very good inhibitors at all for inhibition of class 2 kinases, which are insensitive to wortmannin or PI103. So we were left with molecular approaches rather than pharmacological ones. And what we did instead was to silence class 2 PI3 kinase using siRNA. And what is shown here is a Western blot for the kinase, validating the fact that the siRNA approach almost entirely eliminated the class 2 kinases by 95 or 90%. So under these conditions, we can now inquire whether class 2 kinases are essential for the activity of SUB-P on phosphoinositides. So here's a control, one siRNA, a different siRNA, and disappointingly, the results were the same as before. There's no change in the generation of PI3-4 when we eliminate class 2 kinases. So class 1 kinase is not related, class 2 is not essential either, because this be an effect of SUB-P that occurs independently, completely, of host kinases. And perhaps the cleanest way of investigating this is to use a biological system that just doesn't have PI3 kinases. And that system is the yeast Saccharomyces cerevisiae. So Saccharomyces, unlike mammalian cells, doesn't have class 1 and doesn't have class 2 PI3 kinases. And so we went to yeast, expressed in the yeast the probe that detects PI3-4P2. And obviously, because they don't have class 1 and class 2 kinases, the probe is soluble because there's no PI3-4 to be detected. But if we now transfect or transform and express in the yeast SUB-P, SUB-P generates very nice large amounts of PI3-4P2 on the membrane of the yeast, despite the fact the yeast don't have class 1 or class 2 kinases. The mutant form of SUB-P, which is inactive, doesn't do it. So at this stage, we were totally convinced that it is possible to generate PI3-4P2, whether in mammalian cells or in yeast, by a mechanism that depends on SUB-P, but does not require PI3 kinases. So that's what I just mentioned. And this involves then a totally distinct biosynthetic pathway to account for the formation of PI3-4. What could be making PI3-4 when you don't have 3 kinases? And one possibility we considered was that perhaps SUB-P directly was transferring a phosphate within phosphatidylinositol bisphosphate 4,5 phosphorylated so as to translocate the phosphate on position 5 to position 3. In other words, in addition to being a phosphatase, it may be serving as a phosphotransferase. That's a nice hypothesis, but how do you prove it? And so we followed the dictum of F. Racker that said, don't waste clean thinking on dirty systems. You have to simplify and try to directly assess your hypothesis without the compounding problems of other proteins and other enzymes. So what we did was we generated recombinant SUB-P in bacteria, purified recombinant SUB-P. We also generated pure lipid liposomes of defined phospholipid composition. We mixed the two and probed for the formation, the appearance of PI3-4P2 by adding the recombinant biosensor that I mentioned before with eGFP. In other words, if PI3-4P2 is generated on the membrane of the liposomes, we expect the biosensor to bind to the liposome outer leaflet. So this is just a proof of principle to make sure that we could actually detect binding of the biosynthetic probe to liposomes containing 3,4-P2. So we made giant unilamellar liposomes containing various amounts of PI3-4P2. the substrate of the probe that we've been using all along. And so if you don't have any PI3,4P2, these are inverted images, you can see that the probe is soluble, but none of it binds to the membrane of the liposome. As you increase the amount of the lipid on the membrane of the giant liposomes, you get progressive recruitment of the probe to the membrane. And we know that this is a specific phenomenon because it's not only concentration dependent, but it doesn't work with lipids other than PI3,4P2. PI4,5P2 would not do it. Okay, so now we have a clean system where we can visualize the generation of PI3,4P2. We can add recombinant sub-P in the absence of any other enzymes or kinases or even ATP. So is sub-P sufficient in a cell-free system to generate PI3,4P2? So what we did in this case is we made giant unilamellar liposomes. 3% PI4,5P2 was added to the membrane. And we added the probe that detects PI3,4P2. And you can see that in the presence of PI4,5P2, the probe is unable to bind. If we then add sub-P to inquire whether we can transfer phosphate in position 5 to position 3, we get this result. So addition of sub-P induces in vitro the generation of PI3,4P2 from PI4,5P2 on a membrane that's purely lipid in the absence of ATP and in the absence of any other protein. And the mutant form of sub-P or inactivated form of sub-P is unable to do this. If you use PI4P as the lipid present in the liposomes, you are unable to form PI3,4P2. And these results are summarized here. And the conclusion of these experiments is that all you need is sub-P and PI4,5P2 to generate PI3,4P2. So sub-P suffices to generate 3,4P2 from 4,5P2 in vitro. This raises the question of whether other lipids are also being generated. In fact, this is not so much a question we asked, but we sent the paper for publication, and reviewers, who are always very smart, asked, well, you're using a probe. Can you show this by chemical means as well? And it's not trivial to detect phosphoinositide by chemical means, but there is a group in Cambridge by Phil Hawkins and Len Stevens that have a mass spectrometry method to detect different phosphoinositides. So we contacted them, we initiated a collaboration, and they were willing and able to measure in our purely recombinant system the lipid composition. So again, recombinant sub-P, pure lipid liposomes containing PI4,5P2, and the rest of it is phosphatidylserine and phosphatidylcholine. And lo and behold, when cells are, or the liposomes are treated with sub-P, you see the appearance in their spectrograms of a peak that corresponds to PI3,4P2, which is absence when you use the mutant form of sub-P. And in fact, because of their sophisticated system, they are able to detect the presence of multiple lipids. And so what they see is that sub-P induces the gradual disappearance of PI4,5P2, the formation of PI4 or PI5P, the generation over time of phosphatidylinositol, and all of these are markers of the phosphatase activity of sub-P. But perhaps more interestingly, you also induce the formation of 3' phosphorylated phosphoinositides, both 3,4,5P3, PI3P, in addition to PI3,4P2 are generated. So what apparently is happening is, starting with PI45P2, we are transferring one of the phosphates to position 3, generating either PI345P3, which can then be dephosphorylated into PI34, or directly transferring the 5-phosphate of PI45P2 to position 3 to generate PI34P2. And if you think about it, mammalian cells developed PI3 kinases to take PI45P2 and, using the energy of ATP, generate PI345P3. This is generation of 3' phosphorylated phosphoinositides via a kinase. Bacteria have learned to do something similar without using ATP by a phosphotransferase mechanism, using the high energy of formation of PI45P2 to transfer a phospholipid, a phosphate into phospholipid to position 3. So there are two different, very distinct mechanisms of generating 3' phosphorylated phosphoinositides. One is a kinase, which is a mammalian or a metazoan system, and the phosphotransferase, which is a really unexpected case of convergent evolution. So all the work that I mentioned to this point was by a brilliant student, Dr. Glenn Walpole. He now has his PhD in collaboration with Drs. Brummel, Theron, and Hammond. So this is the first story I wanted to tell you. I'll tell you a second story starting now. If there's any questions, feel free to interrupt and ask me now. And if there's nothing that's imminent, let me proceed with a second related story here. And this also has to do with the interaction between mammalian cells and bacterial effectors. And this is the work of a postdoc in the lab, Dr. Michelle Maxon. And just as a reminder, I'm sure you all know, in mammalian cells, we have different pH in different endomembrane structures. Mitochondria are slightly alkaline. The cytosol is nearly neutral, as is the nucleus and the endoplasmic reticulum. But the secretory pathway is gradually more acidic, such that secretory granules have a pH of 5.5. And conversely, the endocytic pathway starts out as being neutral and becomes progressively more acidic, so that early endosomes are slightly less acidic than late endosomes, whereas lysosomes reach pHs lower than 5. And the low pH of the lysosomes is essential for bactericidal purposes and for degradation of materials inside the lysosome. And as we all probably know, the mechanism responsible in all of these instances for the acidification of the endomembrane compartments is the vacuolar ATPase or proton pump. This is a multi-subunit rotary motor pump that takes protons from the cytosol and uses ATP to drive them across the membrane into the lumen of the organelles by a process that is sensitive to bacillomycin and concanomycin. And actually, the final pH reached by each compartment depends on the proton pump activity, but also on proton leaks and on counterions. And it's been very difficult to assess whether some organelles are more acidic than others because they have more pumps, or because they have more active pumps, or because they have more of a leak, or because they have different counterion conductance. And part of the problem in assessing the density of pumps is that the number of reagents available to quantify pumps in membranes is very poor. So how can we determine if the density of vacuolar ATPases, of proton pumps, is higher in more acidic compartments? One possibility, obviously, is immunostaining, and there are antibodies available and commercially available too for immunostaining the VATPase. Now, these are 3 different commercially available antibodies, and in fact, these images are taken from the websites of the manufacturers of the antibodies. Now, we all know what the lysosomes should look like, or the Golgi should look like. Those are the structures that should be rich in proton pumps. And this doesn't look like lysosomes. This looks like garbage. This looks like the endoplasmic reticulum, which we know is not endowed with proton pumps. This other antibody stains the nucleus and faintly stains the cytosol. So it's not trivial to find antibodies to stain the number of proton pumps present on the organelles of mammalian cells, let alone antibodies that are good enough to quantify the number of proton pumps. So what do we do? In fact, some of these antibodies have been used to claim that oncogenic proteins alter the level of VATPases in the plasma membrane. But when you see these kinds of images and you compare them to the cytosolic staining, I'm sure you will be convinced that these antibodies are not reliable. So VATPase antibodies really suck. They're not good, and I wouldn't trust them to stain, at least not the numbers of ATPases available in mammalian cells. But as usual, evolution and microbes have developed better tools. Microbes know best. So what I'm going to tell you next is based on this paper that was published a few years ago, where they found that Legionella, one of the intracellular pathogens that I mentioned before, is capable of inhibiting the vacuolar ATPase. So how has Legionella figured out means of inhibiting the vacuolar ATPase? And what it does is it generates yet another effector. Again, a protein that the pathogen injects into the cytosol of the host cell. And in this case, the effector has been termed SITK. So what is SITK and how does SITK work? So SITK is a relatively small molecular weight protein that was subsequently found to bind directly to one of the subunits of the proton pump. This probably will give you a better idea. This is a beautiful cryo-electron microscopy determination of the structure of the vacuolar ATPase. This is the transmembrane domain, the rotary component. This is the ATPase part. And right here, binding to one of the large A subunits is SITK, shown in green. It binds strictly, directly to the A subunit and gums up the rotary activation of the proton pump. So the person that developed this beautiful structure and was studying SITK, Dr. John Rubinstein, approached us and said, hey, can we use SITK to visualize the proton pumps? And so together with John, we generated a construct for mammalian expression of SITK as a chimeric construct with GFP. And so this is a HeLa cell, a regular mammalian cell, which has been preloaded with a dextran to reveal the position of lysosomes. So the pink, purple here are lysosomes. Green is SITK GFP, which we were hoping would specifically label proton pumps and thereby highlight the membrane of the lysosomes. And clearly, that was not the case. And I think the reason we are unable to see SITK binding to the membrane of lysosomes here is that we are expressing the protein in such a huge excess that the bulk of it is cytosolic, having saturated all the possible binding sites. And indeed, if you find in the cover slip cells that have very low expression levels, such that the cytosolic level is negligible, you can now start visualizing punctate structures labeled with CYP-K, presumably by binding to the vacuolar ATPase, and many of them co-localize perfectly well with VATPases. So in theory, one can use expression of a plasmid with chimeric CYP-K and GFP to visualize the VATPase. But there are two problems. One of them is that the level of expression is absolutely critical and very difficult to control. The second one is that we know that the expression of Legionella CYP-K affects the pH of the cells, and as such could be affecting the biology of the cells. So if we express an inhibitor and we look the morning after, we really don't know if the biology of the cells has been affected. So do we have any alternatives? And what we decided to do instead is to use CYP-K as a label of vacuolar ATPases post hoc. In other words, we generated a fluorescently conjugated recombinant CYP-K. This is generated in bacteria, purified from bacteria, and then covalently labeled with an NHS ester to a fluorescent probe. That created a low stoichiometry CYP-K labeled with a red fluorescent probe, Alexa 568. So CYP-K Alexa 568 was going to be used to stain cells in a manner analogous to immunostaining. So what we do is we fix the cells, we permeabilize the membrane to allow the entry of CYP-K Alexa into the cells, and then we add whatever amount of CYP-K we prefer to try to stain the cells. And lo and behold, what we find is a beautifully reproducible staining of punctate structures throughout the cell that, at least in principle, suspected would be VATPases. So how do we know that the binding is specific? And how do we know that CYP-K is actually labeling VATPases at all? So specificity was tested by comparing cells that were directly labeled with CYP-K to cells that first pre-treated with unlabeled CYP-K to saturate binding sites, followed by treatment with CYP-K with a fluorescent label. And what you see is that at least the binding that we're seeing is saturable, so that if you pre-bind the VATPases with unlabeled CYP-K, you prevent the binding of the labeled CYP-K. That is suggestive, but by no means proof that what we're labeling here are indeed VATPases. I think the most conclusive proof of the pudding came when we used mutant cells without VATPases. Now, mammalian cells are not viable without VATPases. It's an absolutely essential enzyme. But yeast, again, Saccharomyces, can survive in the absence of VATPases. And the reason for that is that yeast are normally grown in very acidic media and can take up acidic medium into their vacuoles to make up for the acidification normally generated by the pump. And so it's possible to isolate mutant forms of yeast that either do have a VATPase or they don't. And the mutants lacking the VATPase are called VMA1 delta. We then fixed, permeabilized the yeast and overlaid them with CYP-K Alexa. And you can see that CYP-K Alexa beautifully labels the membrane of the yeast vacuole in every instance. But perhaps more importantly, yeast vacuoles lacking the VATPase show absolutely no labeling at all. What's blue here is the nucleus. What's red here is the VATPase labeling of the yeast vacuole. So now that we are convinced that we are in fact labeling VATPases, we can proceed to see what kinds of organelles we have labeled in the mammalian cells. So here's the HeLa cell that I showed you before. You have some labeling near the nucleus and a lot of peripheral labeling as well. So what are they? And to find out the identity of the individual labeled organelles, we used co-labeling and co-localization with specific organellar markers. So LAMP1 is a specific marker of late endosomes and lysosomes. In this case, it's green. CYTK, in this case, is purple. And I'll be showing you throughout co-labeling of a marker with CYTK, and co-localization will be indicated in orange. So you can see that LAMP1, lysosomes, certainly co-localize as we expected with CYTK, and you get very nice co-localization images. The trans-Golgi, which we expected to be acidic, and in fact the trans-Golgi is the most acidic part of the Golgi, is labeled here with a trans-Golgi marker, labeled with CYTK, and there's exquisite co-localization of the two. The cis-Golgi, which is much less acidic than the trans-Golgi, can be identified with another marker, a distinct one, and you can see that even though CYTK is near the cis-Golgi, it really doesn't quite overlap precisely. And the reason is that CYTK is probably labeling the medial and trans-Golgi and not the cis-Golgi. So when you look for co-localization, it's fairly minimal, as is also the case with the endoplasmic reticulum. Here, the green marker is the endoplasmic reticulum, the purple marker is CYTK, and co-localization is missing. And in fact, CYTK seems to be hiding in all the places where the ER is not present. Mitochondria, which we know are alkaline and should therefore not have vacuolar ATPases, are totally distinct from CYTK. You can see that both are very obvious, but there is no co-localization. So together, these experiments are consistent with the notion that CYTK is very specifically labeling those organelles that are lining the membrane of acidic compartments. And just for comparison's sake, this is what you get in our hands when you label LAMP1 with the commercially available antibodies. You can see the commercially available antibody give you this fuzzy, dirty staining that doesn't co-localize well with lysosomes or with the trans-Golgi. Okay. Now that we trust and know that CYTK is a good marker for vacuolar ATPases, we can study real biology with it. And I'm not going to take too much time. I know it's only 5 minutes left to the hour, but here's CYTK labeling of cells. And here we're studying macrophages, which are cells that form phagosomes and engulf particles. And what we managed to do was follow the acquisition of vacuolar ATPases as the phagosome becomes progressively more acidic. So here's a marker of the plasma membrane. When a phagosome just was created, it has plasma membrane component, but no vacuolar ATPases. When the phagosome starts fusing with early endosomes, denoted by RAP5 or by PI3P, it starts acquiring vacuolar ATPases. And as the phagosome continues to mature and becomes a phagolysosome, acquiring RAP7 and markers of the lysosome, it becomes very rich in vacuolar ATPases. So CYTK has proven to be in our hands a very useful marker and very specific marker for vacuolar ATPases, and has even enabled us to do some quantitative assessment of the number of proton pumps per organelle. It's also interesting that the labeling by CYTK is discrete and tells us that when you look at organelles like macropinosomes or phagosomes, you find that the pumps are present in some regions, but not in all. And even when you have tubular lysosomes, you can see distinct subdomains. So it opens up the possibility of studying the suborganellar structure of the vacuolar ATPase, quantifying the vacuolar ATPase in different organelles, and monitoring the fate of the vacuolar ATPase. And all of this work is that of Michel Maxon, And as I mentioned, this is in collaboration with John Rubinstein and Yazan Abbas. So I think I'll stop here. We can switch to Spanish if you prefer. We can ask and answer questions in English. And I'll just try to stop sharing my screen. Sí.
Muchas gracias, Sergio. ¿Me escuchas?
Escucho muy bien, sí.
Bueno, vamos a pasar a las preguntas. Vamos a hacer una aquí en el auditorio y pasaremos a la otra vía Zoom. ¿Alguien?
Sergio, muchas gracias por tu presentación. Mi pregunta es, ¿el papel de AKT se requiere en el proceso de infección para inducir todos esos cambios en el citoesqueleto, para la invasión de la bacteria? ¿O no?
Los efectos más importantes de AKT no son en el citoesqueleto, sino que son en la proliferación, el mantenimiento de la viabilidad y la respuesta inmunológica misma, la inflamación. Hay otros dos efectores de salmonella, se llaman SOP-E y SOP-E2, que son los más importantes en producir el efecto sobre el citoesqueleto, de manera que uno puede eliminar SOP-B y mantener los efectos sobre el citoesqueleto.
Es que me pareció que no ocurrían esos cambios cuando SOP-B estaba mutada, por eso preguntaba.
Lo que no ocurre, sí, lo que no ocurre es el reclutamiento o la formación de PI3-4P2 a los ruffles, ¿cómo se dice ruffles en español? So, el ruffling, las convoluciones de la membrana y la invasión pueden proceder en la ausencia de SOP-B, aunque no tengas PI3-4P2. Es muy interesante porque hay 3 diferentes efectores que pueden producir la invasión cada uno por sí mismo, SOP-E, SOP-E2 y SOP-B. Si uno elimina cualquiera de los 3, todavía hay efectos citoesqueleto y hay infección. Los más importantes son SOP-E y SOP-E2, pero aún sin esos dos, SOP-B puede producir invasión con pequeños efectos sobre el citoesqueleto. Si esos efectos del citoesqueleto dependen de 3, 4 y 2, no sabemos.
Gracias.
Por nada.
Tenemos una pregunta en el chat de Alberto Darson. ¿SITK es pequeña? ¿Se podría introducir a células vivas con poliargininas?
¿Puedes repetir la pregunta para ser seguro?
Sí, comenta que SITK es pequeña. ¿Se podría introducir a células vivas con poliargininas?
Supongo que en principio, sí. No sé qué tan eficiente sería el proceso, pero sí, todas las proteínas de ese tamaño con poliarginina, polilisina pueden entrar a las células. Eso conceptualmente es posible, sí. Sí, la única manera como nosotros tratamos es haciendo transfección, o se puede hacer microinyección también.
Otra pregunta aquí en el auditorio. Voy a hacerla yo, Sergio. Quiero hacerte 2 preguntas. Una es en relación al origen de la bomba de protones en tus experimentos. ¿Son de síntesis de novo o vienen de fusión? Por ejemplo, el fagosoma. Yo tenía entendido, a lo mejor me equivoco, de que la incorporación de la bomba de protones al fagosoma viene de un proceso de fusión con otras organelos.
Sí,
¿Es el caso o viene de incorporación?
Tienes toda la razón. Eso es un proceso de fusión. Es demasiado rápido para hacer biosíntesis. No requiere biosíntesis y lo que indica realmente es primero fusión con early endosomes, después fusión con late endosomes y después fusión con lysosomes. Y corresponde con marcadores de fusión y se previene cuando previenes la fusión.
Okay. Y la otra pregunta es quizás más, me pareció muy atractivo todo el mecanismo de acción de SOFB. Me quiero preguntar no tanto en relación a lo que mostraste respecto a cómo la salmonela utiliza SOFB, sino quiero preguntarte, me da la impresión de que SOFB se vuelve una herramienta para estudiar fosfoinosítidos. No sé si podrías dar algún ejemplo de cómo poder usar SOFB como una herramienta para estudiar fosfoinosítidos.
Claro, sí, cualquier sistema en el que quieras tener aparición aguda de PI340P2, puedes usar la fotoactivación de SOFB. Pero además de eso, nosotros y otros grupos han usado SOFB por su actividad de fosfatasa. Es una fosfatasa extremadamente activa y si la inyecta uno a las células, lo que tienes no es solo formación de PI340P2 y activación de fosfo-AKT, sino que tienes una hidrólisis masiva de PI45P2 y de PI345 si es que existe. Entonces la gente lo ha usado como una fosfatasa muy activa para eliminar fosfoinosítidos de la membrana plasmática y se ha hecho en drosófila y se ha hecho en células de mamífero y funciona extremadamente bien.
Gracias.
Tenemos otra pregunta en el chat de Takuya Nishigaki. ¿Cómo se introdujo la cumarina a lisina específica de SOFB?
Biosintéticamente. Eso es hay un análogo de y es una tecnología muy sofisticada. Hay un análogo del codón de lisina que permite la incorporación de hidroxicumarina de lisina. No me pregunten los detalles, pero le puedo mandar una referencia. Y es una técnica muy elegante porque uno puede específicamente seleccionar una sola de las lisinas en la proteína para usar en esa posición el codón, creo que se llama amber codón, donde puede uno poner el análogo de lisina que se puede reaccionar con la hidroxicumarina.
Una otra pregunta aquí en el auditorio. ¿Tienes más?
Bueno, aquí en el auditorio no hay, pero continuamos con las de Zoom. Pregunta Omar Pantoja, ¿cómo está controlada la inserción de la BATPasa en los endosomas?
¿Cómo está controlada la...? ¿Cuál es? Perdí una palabra.
La inserción de la ATPasa en los endosomas.
¿La activación o la...?
La inserción. ¿Cómo está controlada la inserción de la BATPasa en los endosomas?
Inserción. Inserción, esa es la palabra que no había. Muy buena pregunta. La BATPasa consiste de dos componentes, la parte B0 y la parte B1. La parte B0 se sintetiza y de manera cotranslacional se incorpora desde el retículo endoplásmico. Y después, en algún momento que no está absolutamente claro, la sección B1 se asocia con la sección B0. ¿En qué momento sucede? No está claro, pero muy probablemente al nivel del Golgi o pre-Golgi. ¿Cómo llega a los endosomas y cómo se controla el nivel de BATPasas en los endosomas comparado con late endosomes, comparado con los lisosomas? No está claro de ninguna manera, pero todo viene a través del sistema biosintético, del retículo endoplásmico al ERGIC, al Golgi y a los endosomas.
Y otra más por acá en Zoom, de Alberto Garzón. ¿Cuál es su marcador de membrana preferido?
¿Cuál membrana? ¿Membrana plasmática?
Ahí está, ¿es Alberto conectado?
Sí, claro.
So,
La plasmática, claro.
plasmática. So, depende, Beto, si quieres marcar por fuera o marcar por dentro. Pero si quieres marcar
por fuera.
Por fuera. Ok. Si quieres marcar por fuera, puedes usar un compuesto que se llama cell mask o compuestos que se llaman FM143 o FM464. Todos estos son compuestos fluorescentes anfifílicos que se pegan a la monocapa externa de la membrana y no penetran a la célula, no atraviesan la bicapa. FM es reversible, cell mask es mucho más irreversible. La otra manera de hacerlo, si son células que tienen carbohidratos, es usar lectinas marcadas como wheat germ agglutinin, etc. Todos ellos funcionan muy bien, todos ellos tienen sus detalles y su especialidad. Algunos no pueden producir endocitosis o se transportan rápidamente por endocitosis. Algunos de ellos, no, la fotoactivación le hace daño a las células, pero esos son detalles.
Gracias.
Por nada. Hay manera también, Beto, de marcar a la membrana plasmática con transfección de compuestos fluorescentes, GFPs, por ejemplo, usando KRAS o HRAS constructs o Lin constructs, que específicamente van a la membrana plasmática porque tienen acilación específica.
Gracias. Omar Pantoja quiere hacer un comentario personalmente.
Claro.
Hola, Sergio, gracias por el seminario. En cuanto a la incorporación de la VATPasa, mencionas que el sector B0 se integra en el retículo.
Sí.
Y eventualmente se va a asociar con el B1 para hacer toda la parte catalítica.
Sí.
Entonces, digamos que la síntesis de toda la parte de la membrana podría estar controlando en todo la formación, o al revés, o es la parte citoplásmica, o cómo estaría, o son las dos.
No solo son las dos, hay un tercer componente. Ahora está la gente empezando a descubrir que hay otras proteínas que se pegan a la VATPasa, que no son componentes de la VATPasa madura, pero que parece que regulan la interacción de la B1 con la B0. Entonces, lo que la gente empieza a creer ahora, no hay datos muy conclusivos, como se diga, que se genera la parte B0 y esa puede estar bloqueada por una proteína que no es un componente final de la VATPasa, pero que va a permanecer asociada a la B0 y previene la asociación con la B1 hasta que la B0 llegue a donde debe llegar. Y por esa razón, por ejemplo, no hay bomba de protones activa en el retículo endoplásmico, a pesar de que es ahí donde se genera la B0.
Ok. Muchas gracias, sí.
Nada.
Interesante, gracias.
Y finalmente, Takuya tiene otra pregunta sobre el uso de stop codon, pero no entiendo muy bien la pregunta. No sé si quieres hacerla directamente, Takuya.
Mi pregunta ya me contestó y muchas gracias. Excelente, muchísimas gracias. Excelente presentación.
No hay por qué. Gracias.
Muchas gracias, Sergio. La verdad fue un placer escucharte. Muy interesante todo lo que nos platicamos. Yo creo que de aquí varios grupos se debieron haber aprendido muchísimo de tu plática de hoy. Y bueno, para los que no sepan, Sergio va a estar nuevamente con nosotros en la conmemoración de nuestro departamento. Entonces, pues habrá más oportunidad de saber de tu trabajo. Muchas gracias otra vez, Sergio.
Al contrario, gracias a ustedes. Y sí, para la gente que va a estar conectada el día de la celebración del instituto, la plática que voy a dar entonces es un tema diferente, así que no tienen excusa para no oírla.
Gracias, muy bonita plática, Sergio.
Hasta luego y gracias a ustedes.